identical twins. And so individuals differ for all kinds of molecular reasons. We talked about indels, SNPs, all those kinds of things. Now with whole genome sequencing becoming so inexpensive, lots of whole families have been totally sequenced. So you can see exactly what sort of changes are made every generation in the genome. Each new child, each baby, has 30 to 50 new mutations. And so that's a tremendous amount of variability coming in every generation. This is a large population, the human population, compared to something like the condor or the cheetah. I want to focus for a few minutes on two very important different kinds of mutations and how they play into how individuals change over time. Remember, that's all evolution is. Evolution is change in allele frequency over time. As long as the allele frequency stays the same, then nothing changes. But we know that all sorts of things affect allele frequency. Mutation is inexorable. Mutation never stops. Drift is automatic. Drift never stops. And so change happens all the time. But two kinds of changes are really interesting. I mentioned them briefly on Wednesday. The first is duplications. Duplications are the only reason you're sitting here. Starting off for several billion years, the Earth only had bacteria. And as eukaryotes evolved, as organisms became more complex, yeast and worms and flies, that complexity is driven by the number of proteins, which is only partly explained by the number of genes. And so as life became more complex, we see evidence in the DNA sequence of whole genome duplication. The simple yeast, Saccharomyces, has 2 whole genome duplications. And once that happens, then genes, you have a pair now of identical genes, they're no longer constrained by selection. One is, but one is free to mutate. And that's how we get complex proteins. That's how we got all the globins that allow for placental development. That's how we got complex immunoglobulins, is mutations, duplications within a gene, giving larger and larger genes with more function. And so duplication and divergence is a very strong driver of change. The other strong driver of change are regulatory mutations. You know, we talked about some of the homeotic genes. We talked about antennapedia. And your book goes into a lot more detail on that. And so the difference, the very, very obvious difference in insects, where a lot of this stuff was done, going from a millipede to a fly, the very simple act of turning off the thorax in a certain segment and just calling it an abdomen. Millipedes have legs on every segment, abdomens don't. The difference between a fly, like Drosophila, and a dragonfly, Drosophila has just wings on one thoracic segment. Dragonflies have wings on 2 thoracic segments. They have 4 wings. And so those dramatic differences in what an individual looks like can be brought about by very, very simple mutations. And so all of that feeds into how individuals become different, how evolution occurs just by changes in allele frequency, changes in alleles. Another really important driver of change is transposable elements. You may think of transposable elements as bad because when they jump, if they jump into a gene, they can disable it. But the reality is that transposable elements have driven most of the changes, the large changes, inversions, translocations, duplications, by their transposition, because they break the chromosome when they transpose. Transposable elements are again a major driver of how genomes become different over time, leading again to speciation. We'll talk about that soon. And so transposable elements are really an important component of how individuals, how species become different. So we've been talking about species. Let's finally define what a species is. So a species is a group of individuals that are actually or potentially reproductively isolated. And that's the key, isolation. Yes?
So just a quick question on variability. So variability is due to gene duplication, divergence, and regulatory mutations.
Everything we talked about on Wednesday. There's a whole long list of why individuals are variable. A whole long list of
Oh, so genetic variability.
Anybody else? Yes?
For isolated, does that mean like they reproduce?
Well, so let's discuss that. So this is, again, like the definition that I gave you for gene, I think on the first day of class. This is a terrible definition. The definition of gene is a terrible definition. Why is this definition terrible? Well, because everybody can think of exceptions to this. You know that the wolf is a species and dog is a species. Those are two recognizable different species. And yet you can very easily hybridize those two species and form fertile offspring. So this is one of those species that's kind of okay, but there really is nothing better, just because of the biology. And so what this means is that, and I underlined isolated, and let's emphasize that, because isolation plays into one of those pressures, migration. So we'll pull all this together eventually today, but this whole definition of a species has all kinds of problems. And the problems are evident if you ever read any of the literature from people who look at different species and name species, all the systematists, they can never agree on all the species. What that means is that for a group of organisms, one systematist will say, this is all one species. Another will publish another paper a year later and say, no, this is really two different subspecies, or two different sibling species, or two completely different species. So for a lot of species, there's tremendous controversy over what actually defines whether this individual is in this group or that group. The lines are so blurred. It's very easy. I can put you in a room with a chimp and a human, and you would have no problem making a decision, probably, which group those two individuals belong to. But if I gave you a German Shepherd and a wolf, it might be a little bit more difficult. It's not just looks. And so 20 years ago, people thought DNA was the answer. All we have to do is sequence everything and it'll be obvious. And that's not true either. And so species is another word like gene that is, it's a good word, but the definition is terrible. And for definition that you have for this, there are multiple exceptions. Science cannot agree in all cases on whether this individual goes here or there, whether this is one big species or multiple species. So just keep that in mind as we go through this, that we have a problem with this word scientifically. I cut you off before.
With species, can't you tell if they're within the same species if they can intermate and have viable offspring?
Like a wolf and a dog,
But then the offspring have to be viable themselves.
and they
correct?
are.
And be able to not be infertile.
You're absolutely right,
So then that would.
but they are. The wolf and the dog can make fertile offspring.
Something better than a dog.
And I think that's true. I don't think foxes go like that.
You know, but also, so do all killer whales and bottlenose dolphins.
No, absolutely. So you can come up with all kinds of examples
Those are all whales.
that don't fit in here. So there is no one definition that covers all cases. Because with lots of groups of organisms, scientists haven't decided whether this group of organisms is one large species or should be split into two species or are subspecies or are siblings. All those terms are viable and are used scientifically. There is no easy answer because of the complexity of life. That's all there is to it. Did I convince you?
Not necessarily, but I see where you're going.
No, you're absolutely right. I mean, so there are lots of examples, especially when you have two different groups of organisms that have been separated for a long time. You can make a lion and a tiger. So, yeah, that's just the way it is. But this is all we have. There's nothing better than this, as bad as that is. Anybody else? Any comments on that? You just have to live with it, knowing that it's not a perfect definition, and there are lots of counterexamples to this. All right. So the fact that we have this isolation in here means that as we talk about where species come from, how they originate, there are what are called isolation mechanisms. If species come about because they are reproductively isolated, that means that there must be mechanisms to prevent two groups of organisms from mating. And again, I have this migration here. Isolation means that you have cut off the avenue for migration. Remember that migration is one of the drivers of the allele frequency, an allele frequency change. So let's look at isolation mechanisms. We can divide them into two general categories. The first category is, let's do sympatric first. And allopatric. So sympatric is two species in the same area. Allopatric mechanisms are mechanisms that physically separate species. So what I mean by that, so an allopatric mechanism that physically separates species, the Atlantic Ocean physically separates alligators in the Americas from crocodiles in Africa. That is an allopatric mechanism that prevents migration of alligators to crocodiles or crocodiles to alligators. Yes?
I actually kind of rough example of sympatric mechanisms like is it a post mechanism?
males. So what I mean by, so allopatric, a mountain range separates two species of worms, but probably not two species of birds. An ocean separates species on one side or the other. That's a very, very clear, easy definition. For sympatric, we have a collection of a lot of different processes. Remember, we're talking about preventing mating, and so sympatric isolation mechanisms. One, pre-mating, isolation mechanisms that prevent an individual, it prevents two individuals living in the same area from mating with each other. And so, seasonal maturation. You see this in birds a lot, where two species, two different species from the same group, two wrens, live in the same forest. Species number one comes into estrus early in the spring. Species number two comes into estrus late in the spring, and therefore they do not cross-mate with each other. That's a mechanism to prevent reproduction, to prevent species one from mating with species two. They're in the same area, but they have this seasonal difference, maturation, that doesn't allow cross-fertilization. Once you get cross-fertilization, that species disappears. Anybody have a question on that?
I just if I read what that word says next to seasonal.
Maturation. So the females come into estrus at different times in the season. Behavior. Again, remember, these are mechanisms that prevent males of species A from mating with females of species B. Behavioral mechanisms in the animal kingdom are really important in terms of mate selection. What that means is that the male typically has to do a song and a dance, whatever it is. In birds, a song and a dance is really important. And so the female will evaluate that male. First of all, is the song and dance right? But we're not talking about selecting within a group of the same species. We're talking about the female discriminating a male of a different species because he doesn't even know the right song, he doesn't know the right dance. And so this is a mechanism to prevent a male of species A from mating with a female of species B. Everybody see how that works? It's simple and straightforward. And lastly, mechanical. And so in a lot of, actually insects do this, whales also to some extent, lots of organisms, lots of different groups of species, where the male genitalia will only fit a female of the same species. And so you see very exaggerated male genitalia that are geared for one specific group of females in the same species. A male of a similar species, similar but different, will have a different shaped male genitalia. It simply won't fit. And so, again, a mechanism to prevent males of one species from mating with females of the wrong species. That allows a whole group of different species, but within the same category, to coexist in the same area. without blurring the species line. Questions on that? Yeah.
Well, how do you explain, like, when you get, for example, I've seen this a lot, Canadian geese that come down Arizona, they meet with the white geese down there, and they're saying hybrids, like with orange noses on, or orange beaks on, Canadian geese, et cetera.
Nature's not perfect. For everything I'm saying, there will be exceptions in nature. I mean, so the rule is that organisms only mate within their own species. But there are tons of examples where cross-fertilization is possible. You especially see that now that we're blurring the lines and transportation is so easy. You have exotics coming into new areas that have lost these systems because there is no need for them. So that's where, in terms of wildlife conservation, that's where the problem arises. When two organisms have been separated for millions of years, they don't need prenating isolation mechanisms. They're gone. And so you see those kinds of problems. Anybody else? Any questions on this? Yeah.
So for seasonal, migration, could that include other kinds of temporal differences, like flowering time?
Oh, absolutely. Yes, absolutely. I just gave one example. Yes.
For seasonal maturation, what stops like the male from species one just not just mating the first time, but then doing it again?
Because the male undergoes sperm maturation at the same time as the female. He's just, if he's geared to a late seasonal female, he's just not ready to mate. You got to get the hormones set, and so that has to do with the position of the sun. All that seasonal stuff is locked in, genetically programmed, that male and female are going to come into breeding time at the same time. Anybody else? And we have 3 post-mating isolation mechanisms as well. The first one is gamete mortality. You see this mostly in plants that do broadcast pollination. A flower will release a burst of pollen that just disperses everywhere, so it can land on the same species fine. If it lands on the wrong species, mechanisms are in place that the pollen has to have a signal from where it lands that allows the pollen tube to grow out for fertilization. If the pollen lands on the wrong species, it doesn't get that proper signal, that pollen just sits there and eventually dies. And so, again, a mechanism to prevent species A from fertilizing species B at the most primitive level, at the gamete level. Very, very common in plants that do broadcast pollination, not plants that self-pollinate like mangoes, peas. Any questions on that? Zygote lethality. Where you see this a lot is in aquatic environments like the ocean. Again, on coral reefs, for example, where fish spawn, lots of different species may spawn at the same time, where coral spawns, releasing eggs and sperm into the water column all together. If a sperm fertilizes an egg of the wrong species, typically fertilization will start the developmental process. But what happens is the same thing that we saw in the Drosophila embryo. It is the cytoplasm that has the gene regulators that control the very earliest steps of development. So if species A sperm, the haploid nucleus, goes into species B cytoplasm, species B transcription factors will not recognize species A's genes and enhancers, and so that fertilized egg, that zygote, is just going to die. Whatever the first few maternal divisions happen, that'll be it. The zygote, the diploid nucleus, is never going to be able to develop, and the whole thing just dies. Again, a mechanism to prevent species A sperm from fertilizing an egg of species B. Any questions on these two? And lastly, hybrid sterility. This is the classic story of a donkey and a horse and a mule. So donkey and horse are two classical species, regular species. A donkey can be fertilized by a horse. There's slight differences whether donkey fertilizes horse or horse fertilizes donkey, but the outcome is the same. That you can actually develop that hybrid, but that hybrid is sterile. And the reason is, let me find the numbers. So the horse has 64 chromosomes, the donkey has 62 chromosomes, and because of that, the mule is missing one chromosome. And so that one chromosome is going to mean that that mule is never going to be able to have offspring. Again, there's slight differences between whether the male is the horse or the donkey, and there's a little bit of fertility one way or the other, and I forget which way it is, but you can't start a population from it. And so, again, just a mechanism to prevent species A from fertilizing species B. Here, you allow fertilization, but you do not allow a population of fertile hybrid offspring. Again, all know of tons of examples now in the literature where fertile hybrid offspring are appearing. And the reason for that is that species that traditionally have been separated allopatrically, two species on two different continents tend to lose all the sympatric isolation mechanisms. And so two species on two different continents have no mechanism to prevent hybridization. And when they come together, like a lion and a tiger, like any number of examples of exotic introduced species, that you have hybrids, either one or the other or both will go out of existence and new hybrid species will form. And so that's a real problem in conservation biology. Any questions on that? Yes? No? I only have the one example, I'm sorry. Anybody else? Yes?
So with post-mating isolation mechanisms, would you include in there then like a one-way sort of situation where you have one sex that are mating sex, like how they move, where you have maybe equal for one sex and not hybrid, not the other?
I don't know that system. I just can't answer that.
So some species of humans or something have crosses where they'll only get female offspring and crosses.
Oh, yeah, but again, those kinds of systems tend to be very artificial, where you're taking two species that have been separated for millions of years and have lost these kinds of mechanisms because now they're relying on allopatric mechanisms. And so whenever you introduce species that have been physically separated for so long, you very often get fertile hybrids. Again, that's the problem with worldwide conservation, is that exotic introduced species are causing changes Anybody else? Any questions on that? So where do species come from? The easiest picture of where species come from is allopatric speciation. I can't give you a percentage, but it's the one that's easiest to describe. So there are examples of sympatric speciation. What I mean by that is that a group of organisms living in the same area, there have been a number of interesting examples where those two species break into separate, one species breaks into two separate species groups. What I'm going to show you is an easier example of how a new species comes into existence. Going way back about 40 million years ago, there was a giant continent. There was a giant continent that was South America and Africa. And these two continents have recently broken off from Pangaea, and now we have this one continent. There was an organism there in this continent that sort of looked like an alligator or a crocodile. And about 20 million years ago, we have continental drift giving rise to South America, giving rise to Africa. With an ocean in between. In other words, now we have an identical common ancestor back here. Common ancestor ending up as these two continents drifted apart. And now, over time, and so this is a time line, and so with time, we have mutation, we have drift, and we have selection, but they're different. They're just different because everything is random. What we do not have is migration. And so, over the course of millions of years, this common ancestor gave rise to gators and crocs. Very, very simple, straightforward mechanism, an allopatric mechanism brought about by allopatric physically separating what had started out as two identical pools of organisms. Mutation is random, drift is random, selection is different on the two species, and over time we begin to see differences in those two species until a systemicist sits down and says, they're not the same species. Those are two different species.
Another example using that exact same thing.
Well, there are tons of examples. There are hundreds of examples.
Yeah, there's like there's the monkeys, like New World monkeys versus Old World.
Absolutely.
Rheas versus octopus.
It's the same thing. It's the same kind of mechanism in place. Once you put a barrier in place, once you stop migration, that's the key. No migration, no alleles, and really, it takes a very tiny amount of migration. It doesn't take many individuals, going across back and forth to keep two separate pools as the same species. Does everybody see how that works? So most examples in the literature of how species arose by allopatric mechanisms, but more and more people are finding sympatric mechanisms that led to speciation. Any questions on that? Okay, so again, all we're talking about in terms of this evolution is a change in allele frequency. That's all that happens. There's nothing more at stake. As time goes on, mutations accumulate, drift alters, selection alters. As time goes on, species become more and more different. Now the timeline is an interesting feature. So there doesn't seem to be one mechanism, one timeline to fit all. So mammals radiated very, very rapidly after the demise of the dinosaur. Humans radiated very rapidly just within the last 100 to 200,000 years, even a couple million years if you count some of the ancestors, like the Homo crab. Does everybody know what a horseshoe crab is? So a horseshoe crab lives in the ocean. Its head is sort of shaped like a horseshoe. It's a crab. It's got a hard shell and a tail. It's about that big. Big animal. That's one of the most ancient animals on the planet. There are fossilized horseshoe crabs that go back literally tens of millions of years that look absolutely identical to modern horseshoe crab. Now, of course, we don't have their DNA. You can't ask if the DNA has changed, but then your DNA and my DNA is different a little bit anyway. But the thing is that there's not one model to fit every species in terms of how fast or how slow it changes. The horseshoe crab hardly changed at all. Mammals, humans, horses, whales changed very, very fast over the last millions of years.
Other marine fossils would be like coelacanth,
Yep,
blue colorless.
yep. Anybody else?
What's the word called?
I don't know. You can ask them later. So the first person to actually come up with a very good model for how all this stuff happens, as you all know, is Darwin. Well, he's the famous one. Darwin and his buddy came up at about the same time. But Darwin published first and seems to have primacy. And so we have this Darwinian model of evolution. There are some really important things that Darwin said. First of all, slow change. Change happens gradually. Common ancestor. That's what that is. And he came up with the notion of selection. Now, so Darwin was a contemporary of Mendel. I think I mentioned Mendel sent Darwin one of his papers, and Darwin had no clue what he was talking about. So they actually found Mendel's paper stuffed in the back of a drawer in Darwin's desk, not long ago. It didn't make any sense to him. He had no clue. Mendel's work was discovered in 1900. Of course, Darwin's been controversial from the 1860s. In the early 20th century, after Mendel's paper was discovered, there were geneticists and there were evolutionists. and they didn't get along at all. The evolutionary biologists of that day couldn't see how genetic change could be fast enough to account for evolution. They just didn't see it at all. And so for about 20, 25 years, there was bitter fighting between evolutionary biologists and geneticists. It wasn't until a group of mathematical geneticists sat down and outlined the mathematical framework for change in allele frequency that the science of neo-Darwinian evolution in the 1920s began. And so nobody much believes in the old-fashioned Darwinian theory. So neo-Darwinian evolutionary theory is simply all of this plus genetics. Basically what I've been standing up here talking about for the whole semester is the genetic framework is certainly capable of explaining allele frequency change and how that affects evolution. You have a question? You have your hand up, okay. So, neo-Darwinian evolutionary theory is what people now just call evolutionary theory. But it's nothing like the original Darwinian theory. So we have to teach the controversy. The controversy is really very simple and straightforward. There is another model to explain evolution, and that is called punctuated evolution. Actually, there are two models. I'll give you the third one. Punctuated evolution came about, I think it was about the 1970s, maybe a little bit before that, by archaeologists and people looking at fossils. So Darwin's original model had slow, gradual change. That's really important. What punctuated equilibrium, what the fossil hunters say they see in the fossil record is not slow, gradual change. What they see is no change for millions, tens of millions of years, and then suddenly, as an epoch ends, very, very rapid burst of change. When the dinosaurs were wiped out 65 million years ago, there was an incredible burst of evolutionary change as the mammalian radiation began. And so punctuated evolution, let me raise this just a little bit. Punctuated evolution is stasis, or no change, followed by a rapid burst of change. That's what they see in the fossil record. And so starting in about the 1970s, there was the second school of evolutionary thought. These are two different theories to explain the facts of evolution. Evolution happened. Evolution is just change. Change happens. Two very different models to explain how this change comes about. And I want to tell you, the battles between these two groups of scientists were titanic. I mean, it was bitter. And so what happened is what happens in science when there's a disagreement. Both sides go out and look for evidence to support their own ideas. And so from that, you have an enormous number of new fossils for virtually every walking organism on the planet, filling in a lot of gaps, all the holes in the fossil record. And the two sides have sort of merged together as new data comes in. And the bottom line is, both are correct. There's not one model of evolution that fits everything. And so we have some organisms that evolve very, very slowly, and some organisms that don't change at all, like the horseshoe crab doesn't change at all. There's one third model that's also kind of weak. The neutral theory model actually fits both punctuated equilibrium and neo-Darwinian evolution. What the neutral theory says, mutations happen spontaneously all the time. You're never going to stop that. The neutral theory says the vast majority of mutations are neutral. We know that. Because you have 30 to 50 new mutations in you that weren't in your parents. And these new mutations are neutral. They only become selectable when times change. Like when we spray the mosquitoes with DDT. When there's a drought and most plants die and the drought-resistant plants flourish. When there's an ice age. When there's global warming. All of those things act as instantaneous selectors on what had been neutral alleles now become the greatest allele in the world. Any questions on these models?
What exactly is neutral theory? Like, how do you define it?
That most mutations are neutral. They are not selected for or against until the environment changes. Is there a question?
Yes. What is in parentheses stasis?
No change. So stasis means long periods of no change followed by very, very rapid change, like at the end of the era of the dinosaurs. And you see that in all the geological epochs. Within a geological epoch, very, very slow and minimal change, and then sudden dramatic change. Any last questions? See you on Monday.